I think it'd be fair to say that Michigan Biostatistics is one of the top biostatistics programs in the world. When you typically do surveys on departments of biostatistics, we're always in the top 5 in the US, and where we are between 1 and 5 depends on how you want to weight things. University of Michigan is one of the top public universities in the United States. Ann Arbor, I'm a little biased here because I've lived here now for almost 40 years. I think is one of the nicest college towns I've ever experienced. It's a really good place to live. And for any of you for whom it's relevant to raise a family. We grew 3 sons here. We have 2 grandkids here. It's a great place for families. We have a department with a really nice, caring, collegial, collaborative environment, and one in which great biostatistics, great statistical science is done. So it's a really good place to be. And we have a very deep commitment to our research, to our teaching, and to diversity within the department. It's frequently a question, what do biostatisticians do? And you'll be hearing a variety of answers to that over the course of the morning and through the lunch. But here's my version. Biostatisticians develop statistical designs and methods and computational tools for the analysis of biomedical data. And in fact, biostatistics can be thought of as applied statistics in the context of biomedicine. A training you get could equally well be used in other areas. But most of our students, graduates, go on into work in biomedicine. We apply these methods to answer questions in medicine and biology and public health and lots of other disciplines. It's an ideal use, from my perspective, of a background in mathematics or computer science or statistics. It's also a great application if you like biology, if that's your background, or biochemistry or some other such area, but you really like the quantitative side of things. I always knew I loved science, but I always knew I was really crummy in the lab. And so when I found out that I could do a combination of using my math and statistics and computing to allow me to be a scientist, that was incredibly exciting to me. And that took me from a place where I don't know what I'm going to do with my life to this is a really good direction. It didn't solve for me totally the question of what do I want to do with my life. I'm sort of still thinking about it even at age 67, but it made a direction for me that seemed really good. And I think that kind of realization has hit a lot of the people involved in our field. The kinds of things we do are things like trying to understand the basis of human health and human disease, to develop new drugs and decide whether those drugs work and for whom they might work best, to predict how long a person with a particular illness might survive. So these are just a few examples of a myriad of the kinds of things we might do with biostatistics, because honestly, anytime you're dealing with human data, and that's what most of what we do in biostatistics, you're dealing with uncertainty, not lots and lots of information, but more information. How do we make inference in the face of uncertainty in the context of human studies? So as one example of what biostatisticians do, I'll tell you what I do. I develop designs and methods and tools for the analysis of human genetic data. That's my primary focus and has been for 40 years, actually. I apply these methods to understand the genetic basis of human health and disease, with particular focus on type 2 diabetes and related traits like glucose and insulin and obesity. And I and students working with me, staff working with me, and colleagues around the world have identified thousands of places in the human genome that are associated with risk to type 2 diabetes or variability in diabetes-related traits. I also direct our Center for Statistical Genetics and our Genome Science Training Program. If you might be interested in pursuing statistical genetics or genomics as an area, I'd love to talk to you about it. A little bit now about the department. We are, by biostatistics department standards, a relatively large one. We have about 260 students. We have 42, actually, I'd say it's 45 faculty at this point, but you can argue about who gets counted based on the different choices. We have a large number of alumni. We have a lot of research going on in the department. We're part, as we've talked about, of the University of Michigan with more than 50,000 students, more than 6,000 faculty, all here on this one Ann Arbor campus. So quite a large university with all the advantages that a very large institution can provide, in the sense of so many different things to do, so many different things to learn about, so many different people to collaborate with, depending on what you decide your primary interests are. There are probably people at Michigan with whom you can do that. You also have the advantage, even though this is a large department, as departments of biostatistics go, it's still a relatively small group of people. And so one in which you can really expect that you're going to know everybody in your cohort, and you'll know many of the faculty. And so you have some of the advantages for those of you who have been in a small college and liked it, by having this relatively small group within this relatively large university. Well, this very large university, there's no relative about it. You have the advantages of big and small at the same time. Biostatistics is part of the School of Public Health. It's one of the required departments to have a School of Public Health, along with environmental health, epidemiology, health behavior, health education, health management and policy, and human nutrition, and the other departments in our school. And there are about 1,200 students and 200 faculty in that school. This just gives you an idea of where our students are from. The two largest groups by far are from the US and from China, with other numbers from India and South Korea, Taiwan, and elsewhere. We have a very international feel to the department, and one that really welcomes and encourages people to come from all over the world. One of the things that my student recruitment is working on, student recruitment committee right now is working on, is to try to bring people from other countries around the world that haven't come to us yet. The two Ghanaian students are an example of that, people who are coming from other countries, which we'd really like to be seeing happen. Which is not to say we don't want people from China or India or the US, we certainly do. This is sort of a silly slide, but it is a slide that shows you, these are the students in our program. And if you look carefully enough, maybe you can or maybe you can't, you can see there's a lot of different people doing a lot of different things. You can't really see the things they're doing, but there's certainly a lot of different people from a lot of different places. We also have faculty from lots of different places. Listed down below are the different countries. We have about 45 faculty, as I said. Those are on the left, who are our primary faculty, who have primary appointments in this department. And then on the right, we have faculty who are adjunct appointments, or in the lower right, people who are emeritus faculty. So most of the people you'd see most of the time would be the 45 people on the left, with some of the folks on the right, particularly the upper right, around as well. Ann Arbor really is just a cool place. I just like spending time here. My wife and I both grew up on the West Coast, and someone had told us growing up that we'd end up living in the Midwest. I simply would have laughed at them. Which is not to say that the Midwest is bad or anything. It's just the West Coast is where we're from, and we spend a lot of time traveling back and forth there because we have family there who we want to be with. But we've had multiple chances to go back west and thought about it seriously, mainly for family reasons. And in the end, decided each time to stay in Ann Arbor because it is such a good place, a great university, a really good department, and so lots of opportunities. So, in terms of our department and its mission, well, we want to provide high-quality training to the next generation of biostatisticians. That is a raison d'etre for the department. That is why we are here. We want to carry out impactful biostatistics methods research to develop new statistical methods and tools to allow scientific questions to be answered and addressed and answered. We want to bring our biostatistics design and analysis expertise to a wide spectrum of health-related issues. And we want to enhance diversity and equity and inclusion in the data science workforce, with the ultimate goal for all of these things to use data to improve human health. So a little bit more about departmental characteristics. We have very active mentoring in the department. Every one of you who decides to join us will have your own academic advisor. If you work with someone as a graduate student instructor, or particularly as a graduate student research assistant, that person will probably be your primary advisor initially. If you go on and do a PhD, you'll of course have the PhD advisor, and perhaps also another person as a GSRA, a research assistant advisor. And I personally really recommend the model where you have 2 advisors on your dissertation. I think that can be really helpful. I think it's good for kids to have 2 parents. I think it's good for students to have 2 advisors because you learn different things from them. And speaking selfishly as an advisor, it's just more fun having more people contributing to different things. We have a strong focus, as I said before, on diversity, equity, and inclusion and student well-being. We have a really strong computing infrastructure, both in terms of hardware and also in people to help you work with that infrastructure to make it useful to you. We have lots of activities outside of classes. We, of course, have lots of classes, but in addition to classes, we have a seminar series. We have multiple journal clubs. We have multiple working groups. We have journey lectures, and I think I have a slide on that a little bit, where a faculty member talks about how they got to the point they are now, talking about their science, but also about their personal experiences. And then I don't think you should pick a department because it has good parties and good meals and good stuff like that. But if you're picking between departments, pick a place where those things happen, because honestly, good food is good, but you're building relationships. Part of what you're doing as a graduate student is building a network that will potentially last you for the next 4 years. I still have really good friends who I knew from graduate school. That's where I got to know them, and we still interact on a regular basis. That is an important part of graduate school, and the free food is nice too. Just a little bit more on inclusion and wellness. Chidima is a staff member who works both in biostatistics and epidemiology, whose focus is providing support for students experiencing challenges during graduate study. We all experience challenges of various sorts at various times, and she is a resource who can be helpful in a whole wide variety of ways. The university has support as well, but having Chidima directly, relevantly right within the department, I think is a very nice thing. In addition, I mentioned before, we have strong computing infrastructure, but also really good people working on the infrastructure with us and helping you as a student or I as a faculty to use the computing resources well and wisely. We have support for our cluster, supporting large computational jobs and Linux computing, and how to use these high-performance clusters. We have another staff member who does R package development to support maintaining integrated website of the department's software tools, and also taking tools that you might build and helping you harden them so they can be something not just that you can use, but that can be made available to people around the country, around the world. Because honestly, that is an important way to have impact as a biostatistician, is not just to develop methods, which is really important, not just to apply them, which is really important, but to have those tools be useful to other people, because that's a way to extend your impact. Here's information from the software page. You can see we have lots of different software represented. And I'll just leave that there as that. I mentioned journey lectures before. This is something I really like that the department does. I gave one several years ago because I've been here so long. But every semester we'll have 1 or 2 faculty basically talk about where they came from, what have they done, how did they get here. And that kind of information I think is incredibly valuable, because I think it's very easy as a student to think, well, I don't know what I want to do, and nobody else has that problem. And how does what I'm doing relate to what others have done? And so seeing that, you know, people who are pretty successful and doing well in the field, to see what they went through to get to where they are, to see the different kinds of steps they took, missteps maybe that they took, I think is incredibly useful. We also have a lot of student-led activities, which you'll hear more about from the students, but just as a heads up, we have a graduate student seminar, we have a journal club and a brown bag. We have the Biostatistics Student Association, which is run by students, that is largely a social activity. So encouraging people to get involved in a wide range of activities. I'd encourage you to ask students about that. We also have another student activity called the Peer Mentoring Committee, which started out a number of years back so that new students would have a current student as mentor. And we still do that. And that's still an important function of the Peer Mentoring Committee. That committee also took upon itself a number of years back to say, we would like to provide input to the faculty in terms of how we can make our excellent program even better. And so we've had, I'm not sure how many rounds at this point, 3 or 4, I guess, where the peer mentoring committee has gone around to different students, talked about what are things we would like to see improve in the department, things that might change. And we've had incredibly useful interactions where most of the ideas that have been suggested, in fact, have been acted upon and made the department better. StatCom, or Statistics in the Community, is a pro bono statistical consulting service that many of the students take part in. It's an opportunity to give back, to use your growing statistical knowledge to help people in the community who might need it. And then there's also a student diversity, equity, and inclusion committee, just as there is a departmental one. Okay, how many of you are rising freshmen, sophomores, or juniors, not rising seniors? Oh, sorry, how many of you are freshmen, sophomores, or juniors, not seniors? Okay, so for those of you who are not yet seniors, something that you might want to consider is our Big Data Summer Institute. It is a program of about 40 students each year, 30 to 40 students each year, 6 weeks in the summer, focused on big data, focused on 3 or 4 different application areas for big data. Genomics is often one, neuroscience has been one, imaging has been one, electronic health records has been one. And what you'll do is you'll take classes in the morning, and in the afternoon, you'll work in small groups on research projects. Great program, something that if you are thinking of biostatistics, haven't decided, and you're still relatively early in your undergraduate experience, it's something I'd really recommend you consider. This slide is sort of my least favorite of the ones I have, because it just doesn't say enough. Because to say enough, we'd have to have a slide that was as busy as the one with all the students, and you couldn't see that one, so you'd have an even harder time with this one. Students from our program go all over the place. We have lots of our graduates who go into academia, particularly PhD graduates will go choose faculty careers. But academia and other research centers are common for both our master's and our PhD graduates. And there's a few of the places that people have gone in the last couple of years. We have lots of our students going into industry. The drug industry is a major employer. Biotech is a major employer. But just about any area you might think of, there's a need for statistics, applied statistics, biostatistics. And so you see a list of places there. And then we have a lot of people going to government as well. Again, anytime you're dealing with uncertainty, anytime you're trying to summarize information, you need biostatistics. And finally, let me end with a slide that is actually a quotation from our dean, Boehnke, who in fact was a student in our department a long time ago. I'm old enough that I recruited him to our master's program. He then went on to North Carolina for his PhD because he wanted to work in imaging. And at that time, we didn't have any faculty members doing imaging. We now do. But I love this quotation because it talks about what makes public health and biostatistics different from some other fields. We really do believe in compassion. Our work is born from compassion, leading to knowledge and research and action. We believe in innovation. Through creative problem solving, innovative thinking, we lay the groundwork for a healthier world. We believe in inclusion. We work diverse talents across campus and across the world to bring more than ideas. We partner to create lasting solutions. And we pursue impact. And to me, this is incredibly important. And one of the reasons why I am so glad to be in biostatistics is I can imagine the work I do having impact on humanity. And so the ultimate goal of our work is to create positive change and have a lasting effect on health. And so I'll stop there and say, questions, please. Thank you very much.
Would you please use the?
Oh yeah, there are microphones,
Okay.
and if the green dot is on, that means it's working.
No, you'll have to press the button and it'll turn to red.
Okay, so you
Okay,
press
so
the button.
does this work?
Green turns to red, and that means it's on.
You can press it once and there you go. Yeah.
That's counterintuitive.
could impact when you get your research, diabetes, they can probably just take it online.
So, you know, I can't do it myself. You know, identifying thousands of places in the genome associated with human health and disease, that by itself is not going to save any lives. On the other hand, if I can suggest, and I have suggested, multiple targets to drug companies, this is something that you might want to take advantage of in terms of identifying the next diabetes drug. That kind of work is done all the time, and there are successes in that. So yes, I think that's helpful. I also, you know, the work we do can help define risk to particular diseases. So we can use the information we have on the genetics to help identify people at higher or lower risk. Diabetes is harder because there's so much going on with things that have nothing to do with genetics. But cancers, for example, perfect example of where genetic information can truly give substantial information about risk and also about what might be good therapies. So I feel like, yes, the answer is for me. And I think most of our people in the field really believe that as well. Yeah.
Awesome, thank you.
Mhm.
I just want to test these because I think the green might mean that they're all on and the red means they're off. So can you hear me on Zoom? Is this better? Yes. Okay. Everyone turn yours to red, please. And when you talk, turn them to green. Thank you.
Thank you. That makes a lot more sense to me. Yes, I don't. Okay, who else has a question? Please. And pushing the button will turn it to green, maybe. Go ahead.
Do you guys work closely with like the other departments in the School of Public Health, like epidemiology, nutrition science? Like,
Yeah.
do you
So
create
the
like
question was, do we collaborate with other departments in the School of Public Health? And the answer is absolutely yes. Every one of those departments, we have people in our department collaborating with one or more people in those departments. The collaborations with epidemiology are particularly strong. My own collaboration is my wife's an epidemiologist and her office is one floor above mine. But yeah, it's very active. Also with environmental health, there's a lot going on there, but all 6 departments, there are active collaborations across, and not just biostatistics in those, but across all the different groups. There's also a lot of collaboration between us and the medical school. That would be probably our single largest sets of collaborations, just because the medical school is very large, and there's lots of opportunity for doing good work there. Institute for Social Research is another place we have a lot of collaboration. In fact, several of our faculty are jointly appointed between ISR and the department. The Survey Research Center there is one of the largest and best organizations of its kind in the world. And part of the reason for that is they have really good biostatisticians working between them and us. Yeah. So there's lots of local collaboration and lots of collaboration around the world. Yeah. Okay.
Did this program used to have like, dare I say it, like a dual degree with epidemiology in the past? And I wondered why you guys no longer had that.
So I don't think we've ever had a formal dual degree. Actually, when Betsy, my wife, Betsy Foxman, and I first joined the university, we thought, well, gosh, there ought to be a joint a dual degree program. We actually wrote one up, but for a variety of reasons, it didn't happen. As a biostatistics student, you can certainly take a fair number of epidemiology courses in what we used to call a cognate or a minor, but there's still the opportunity to include somewhere between 9 and 12 units of your classes in a different area, and that could be epidemiology if you choose that. We've also had a decent number of students go from a master's in biostat and then do a PhD in epidemiology. And so that's a pretty typical kind of thing to do, not as typical as going on in biostatistics, but it's not an unusual thing to do. And we often have the same thing happen between us and bioinformatics, to start with us and then do a PhD in bioinformatics. Yeah. But no, we don't offer that specifically. I can't hear you. I'm sorry. I'll just get closer. I have old ears, I'm afraid. Yeah. Yeah. So the question was, is there an opportunity for students to interact with, collaborate with people in industry? And the answer to that question is yes. There are quite a lot of students who will do internships and spend their summer working in an industrial setting, most often summer. We also have representatives of industry come and talk with us, to talk with the students in particular, sometimes to recruit, sometimes to just give an idea of what it's like working in industry. And we have people come on a regular basis for those kind of interactions. Other things I should mention in that regard? Local folks. Those would be the most obvious ones from my perspective. Yeah. And honestly, we do also have some collaborations funded with industry. I've had those a time or two in the years, and I'm not alone in that. So that's certainly something that does occur. And so, for example, right now, I don't have something funded by industry, but my work in metabolic diseases, where we have data on metabolites, we have folks from Pfizer who are very actively involved in the collaboration with us. Yeah. And again, I'm not alone in that regard. Yeah. Other questions, please. Maybe this will be the last one.
Yeah, do I need to step here? Okay. So I noticed that U of M also have a bioinformatics program. I know they're like completely different fields, but I'm just wondering, could you provide some insight into like what the similarities and differences between these two programs?
Yeah, I don't think they're completely different fields, just in the sense that biostatistics and epidemiology can be thought of as sort of 2 points on a continuum. I think the same is true for biostatistics and bioinformatics. Our bioinformatics program is a really good one. I was one of the people who helped set it up in the first place. I don't play a large role at all at this point. In fact, no role at all, except having a lot of friends there. In bioinformatics, there is an even stronger commitment to computing than there is in biostatistics on average. These are overlapping distributions. There's less strong commitment to statistics, although there are a lot of people that are very focused on statistics. So it's really, there's a lot of similarity in what we do, but we in biostatistics are very focused on the statistical inference portion of what we're doing, as well as the computing. The computing tends to be perhaps a little secondary, because we use it as a tool to get to where we want to go. But there's a lot of overlap as well.
All right. Hi, everybody. I am Jean. I have a little about me slide. Move this so you can see my face, which you can also see here, but here it is on the screen. So I am an assistant professor in biostatistics. I've been here since 2020. I do also statistical genetics research like Mike. Although there are many people here who don't do statistical genetics research. That said, it's a very cool field. And if you're interested in StatGen, there's a really fabulous group of people here. One of the just like best in the country groups of StatGen folks are here. So I do methods research, and I'll tell you a little bit about the research that I do in the next couple slides. I also teach our causal inference class. So if there's something in here that like piques your interest in the causal inference realm, you can have that to look forward to come and take that class. And sometimes I teach our sort of master's capstone writing class as well. And this picture, since it's a weird one, I'll tell you, I got into vermicob hosting this summer, and I was very excited about how fabulously my worms were doing. So that's that picture. Are we not sharing? That's all right. Yeah. Okay, so hopefully now everyone can see slides and hear me, both in the room and on Zoom. Okay, so I'm going to tell you a little bit about my research, and I'll also tell you that there's too much content in these slides, so we won't talk about all of it. So it's very hard to resist the temptation to just put formulas in slides, even if that's not totally the point. So let's see if I can make these slides visible for you. So genome-wide association studies are kind of the bread and butter of statistical genetics. And I actually got into statistical genetics, I think about 15 years ago is when I first started working on this stuff. And at that time, GWAS, as we call them, were like at least as far as my view of the field, the main thing that we did. And we worked really hard to do them well. And basically a GWAS is you collect a bunch of people out of your population and you measure their genotypes. So that's this X matrix up here. And you measure some phenotypes. So do they have heart disease? What's their blood pressure? Something that you're interested in. And then you just calculate an association between each of their genotypes and that phenotype. And at the end, you get out these coefficients that represent how strongly is that genotype associated with the phenotype and some standard errors. So at that point, for me, 15 years ago, we then make a plot like this. So this is called a Manhattan plot. It's called that because it looks like skyscrapers. And what we're plotting here is each of these dots is one of the variants that we measured. And on the vertical axis, we are plotting how strongly is it associated with that trait. So this is coronary atherosclerosis, heart disease, and we can see some of these positions in the genome are very strongly associated with heart disease. So in 2008, when I started working on this, we would then stop and publish this picture and say, okay, you figure it out now to the biologists, right? So at this point, you actually still don't know why people get heart disease, right? You know, there's all these points in the genome that increase or decrease your risk of heart disease, and we don't know why. And so that is one of the big puzzles of statistical genetics, is taking these associations and trying to figure out, why is this variant associated with heart disease? What biological process is it doing? And does that tell me something about how I can treat heart disease, how I can prevent heart disease? What might happen if I give you a drug that, say, targets this LPA gene? Is that going to make your heart disease better or worse? Is it going to do something to some of your other traits? That's a lot of what we're trying to do. It's at least a lot of what I'm trying to do. People statistical genetics is actually quite broad, and people are doing other things as well, but that's sort of a lot of it. So one of the ways that we can think about how do I understand what these variants are doing is I can take this variant. So here I've picked out this RS and then a long string of numbers. That's a name of a variant. And I picked this variant because it was strongly associated with heart disease. And then I looked up in a database, in this case, I used PhiWeb, every other trait in that database that that variant was associated with. So I can see, okay, well, it's associated with coronary atherosclerosis. That's how I found it. But it's also associated with gallstones and lipids. So I could look at that and think, all right, maybe this variant has something to do with lipids, and maybe the lipids are related to heart disease. I'm not totally sure how that relates to gallstones. Maybe if I talk to a doctor and they can tell me how are lipids or heart disease related to gallstones. But this tells me there's some like common biology between these traits. So a lot of my research is taking that observation that variants share phenotypes. So I'm going to call these CPAs in this talk, cross phenotype association. So phenotype is trait, and then association is what you think it is. So why do these happen, right? Why do I have a variant that's associated with both heart disease and gallstones? So there's a couple things that could be happening. So this drawing is just a little cartoon of the genome, and we have the star, which is the variant that causes some trait, and then the little dot is maybe the variant that I found in my study. So it's not necessarily the same. Maybe it's right next to it and highly associated. So there's things that could be happening, right? I've found that my star variant is associated with both trait 1 and trait 2. It could be that trait 1 causes trait 2, right? So maybe lipids cause heart disease. It could be that there's some common underlying biological regulator, right? Maybe something related to lipids and something related to heart disease are co-regulated. It could be that the variant just does two different things, right? Some variants do participate in lots of different processes, or it could be that I actually sort of made a mistake in that the variant there's just two variants next to each other in the genome. They're doing different things, but they're correlated because they're next to each other. So there's all of these different options for why I could be seeing this. And my goal in my group is to figure out which of these explanations or which combination of these explanations fits best with the pattern of cross-phenotype associations that I'm seeing. So we've approached this in a couple different ways. So that you can kind of take this picture and think of like 3 different things that you can do corresponding to like the different columns of the picture. So one thing that we can do is try to take cross-phenotype associations from lots of traits and see if we can characterize unmeasured biological mediators. So this sort of top picture, we have a common factor that's affecting multiple traits. Can I identify like an underlying mediator network from my patterns of genome associations? The other thing I can do is directly measure causal effects. If I think there might be a causal effect of lipids on heart disease, I can actually test that using only genome-wide associations. And then the last thing I can do is I can look for signals of confounding. So we like to pretend that all of our genome-wide associations are perfect, but sometimes we can get residual confounding, and sometimes that can leak over as patterns in how my beta hats differ slightly from what I might expect them to do. So I'm not going to actually talk about that today, and I'm probably not going to talk too in depth about any of these, but I'll give you an idea of how these work. So I'm going to talk a little bit about these last two, characterizing unmeasured biological mediators and measuring causal effects. So the idea for this, how do I detect mediators project is, all right, so on the left, I have 3 traits that I get to observe, and I've done GWAS for my 3 traits. And the truth is there's 2 underlying biological mediators, and the genetic variants that I find that are associated with these traits can either act through the biological mediators or they can act through some other pattern. So I can ask, how would this network structure appear in the patterns of those beta hats that I get out of GWAS? So if I sort of do some algebra and I assume that some of these effects are linear or like sort of linear, I get to this pattern on the right, where the variants that are associated, let's say, with this factor 2 are going to have effects on all of the traits, right? And the variants that are associated with this factor 1 are just going to have effects on those 2 traits that factor 1 is affecting. So I can look for these patterns. So that's basically the idea, is I take this matrix, right? So I've represented here a matrix as a heat map, and I'm going to decompose it into components that I think represent biological mediators. I'm going to skip the matrix multiplication. So I'll just tell you that we can do this, right? So we have a couple different factor decomposition methods, and we think that we can understand which ones are more likely to represent biological factors and which are just sort of like mathematical patterns. So we take this method that does this like specialized matrix factorization of these beta hats, and we're going to apply it to some risk factors for coronary artery disease and type 2 diabetes. And our goal here is to decompose the heritable component of these traits into components from these underlying biological mediators. So this is sort of our results here. So we've got down here, we have a handful of like obesity related traits, obesity, BMI, waist to hip ratio, basal metabolic rate. We have some hypertension related traits. We have some lipid related traits. And then up at the top, we have type 2 diabetes and coronary artery disease. So these numbers are telling us what proportion of the total heritability for this trait is explained by this factor. So this first factor is clearly explaining a lot of the heritability of BMI and a little bit of the heritability of these obesity phenotypes. And it's explaining 29% of the heritability of type 2 diabetes and 11% of the heritability of coronary artery disease. So doing this, I can sort of see, all right, there is a shared genetic component between these 2 complex diseases, and it's shared also with these obesity related traits. And I can do the same thing for these next factors. Sort of the second one is pretty clearly a hypertension related factor, and that has a much larger contribution to coronary artery disease than it does to type 2 diabetes. And then there's this lipid related factor that explains a small percentage of the heritability of CAD. So that's sort of one example of the results that we get out of this method. There's a couple other like interesting things that we can do with it that I won't get into here, but that's sort of a picture of what those results look like. So the other side of this coin, all right, so I can estimate these mediator networks, but I also told you I can test causal effects if I think there's a causal effect to test. So that strategy is something called Mendelian randomization. And basically the idea here is if I think X causes Y, then any variant that causes X or changes X should change Y in a proportional way. So that's the idea. So if we think back to this matrix pattern, basically I have the pattern where anytime I have a dark color for T1, I should also have a dark color for T2. So this pattern, not consistent with a causal effect because I have some effects on T1 that don't affect T2, but I can go and look and say, is this consistent with a causal effect or not? So that's the idea of Mendelian randomization. I'm going to skip this slide. This one's here for people who are maybe already know instrumental variable analysis. So the strategy with Mendelian randomization is I basically just do a regression testing that pattern that I told you, right? So here I've plotted associations with LDL cholesterol on the horizontal axis and associations with coronary artery disease on the vertical axis. So each of these dots is a genetic variant. And so what I can see is, well, a lot of the variants that have big effects on LDL also have effects on coronary artery disease. These tend to be correlated, right? They're positive. If the variant increases your LDL, it generally increases your CAD risk. It's not across the board the case, right? We have some variants down here that increase your LDL a little bit and actually decrease your CAD. But if I combine the evidence across all of these variants, these are suggesting that increasing LDL is going to increase your CAD risk. So that's the idea of MRs. We take all of these variants and we just ask, what is the consensus opinion about the effect of, in this case, LDL on CAD? And I'm going to skip the details about why is this hard and what is there. There's lots of interesting math to do once you start actually figuring out how do I fit that estimator. And I'm just going to tell you about sort of two interesting extensions that we can do from that. So Mendelian randomization is estimating the effect of one exposure on one outcome. And there's instances where we actually want to estimate the effect of multiple exposures on one outcome simultaneously. And that will allow us to get better estimates of those causal effects. And it allows us to be a little more robust to violations of some of our assumptions. So that's multivariable MR. This is also sort of like an interesting mathematical problem for a bunch of reasons. There's some like cool problems with like nuisance parameters that we have to deal with. If you like Bayesian statistics, this is a good opportunity to do that. And then the second extension that I'm particularly excited about is network Mendelian randomization. So you give me a set of 100 traits and you say, I think these are connected by some causal network. And then I go off and I try to both reconstruct the network and tell you the size of those causal effects. So this is brand new work that we've started working on in my group, and it's, I think, pretty cool. And this is, I think, especially cool because this is extremely hard to do in observational data. This problem is sort of computationally very, very hard, even if you don't have any confounding in observational data. But actually, there's some tricks that we can use via the Mendelian randomization route that make this problem, I'm not going to say easy, but easier or doable at scale, which is pretty cool. So that's my whirlwind overview of cross-phenotype associations for biological discovery. There will not be a test on any of those things. But I wanted to give you like a flavor of the kinds of problems that we're thinking about. Happy to take questions, and I'll be here at the like lunch hour also. So if you want to ask questions, then we can do that.
What is the hardest challenge in terms of researching within the biostatistics field, and what are some of the tools that you think is the most important to confront those challenges?
A good question. Hardest challenges. So that's, I mean, that's going to be different for every field, right? So for a while in statistical genetics, maybe some of the hardest challenges were like computation, right? That like, I want to do this fancy statistical method, but I need to do it 4 million times and I need it to not take a month to do it. So, you know, computation is a challenge, but I often view that as like, if you've gotten to the point where you have a method that works and computation is the problem, you can probably solve that problem, maybe, unless under some circumstances. So that's one thing. I don't know if that's the kind of answer that you were looking for. Yeah, it's a good question. It probably has like a long answer. I'm trying to think of I don't want to just tell you like a list of statistical techniques because that won't be interesting. So I guess I will say I have, like in my work, I use a combination of Bayesian statistics and techniques out of like computer science. So actually like a lot of techniques that were developed for stuff like neural networks are actually really useful for fitting the kinds of problems that we fit. But that said, I think like in biostatistics, there's such a wide array of interesting problems to work on that people I think come at from two angles. Some people come in and say, I care a lot about type 2 diabetes. I'm going to do everything I can to understand type 2 diabetes. And then other people, maybe people like me, not that I don't care about type 2 diabetes, but I came here from the angle of, I like working on these kinds of math problems. This is a really interesting problem to me. You know, how do I solve this like mixture model, or how do I solve this empirical Bayes problem? And then happily, this is like useful. And so you have this combination of how can I do a problem that I think is interesting in a way that's useful? So you can kind of go from either side. If I need to solve this problem, what tools do I need? Or I like this tool, what can I hit with this hammer? I don't know if that answers your question, but yeah.
Can I ask a question?
Yeah.
When you're doing the regression, I was a bit curious. If all the data points are on the far left, how can we use that to make inferences on the far right where there's a couple outliers?
So that's a good question. So in that regression, there's a couple issues. One, we don't like that there's outliers in that regression. So in Mendelian randomization, the fact that there's outliers tells me that at least some of these variants are violating my assumptions. So one of the things that we actually do is we look at that plot, and there's some like ways that we can quantify, are there outliers? And then we say, how much do we believe this slope? And the main thing that we're trying to get out of that regression is the slope of that dotted line. And there's also some weighting that was hidden in that plot, which is that not all of those points sort of contribute equally. But there is a whole bunch of estimators that like try to downweight outliers or try to remove outliers or try to decide which variants actually are outliers. It's a whole interesting field of MR and like sort of generally statistical methods. There is a lot. Let's talk later. Yeah, it's a really cool field, but yeah, if you're interested, I'm happy to give you some stuff to read. Yeah.
Perfect. Should I start or wait for? Hi, everyone. My name is Soumik. I'm a fifth-year PhD student. I've been in this department since 2019. I came in with a master's, and I also got a second master's in biostatistics while I was working on my PhD. And I'm going to be talking a little about stuff that I've been working on for my thesis. Mostly this is work from my first thesis chapter. Although if you want, I can talk in detail about the other work that I've been doing as well. With that, I'd like to start. So as you can see on screen, you'll see some words that you're already maybe familiar with, some which might be new to you. And the key words in this context are: one, Can you hear in the back? Okay, perfect. Yeah, sure. All right. So the keywords you can see in this context are association. You'll see mutual information being mentioned a lot, and these are primarily from context of statistics and information theory. We'll also see a gene network reconstruction as an applied example. Parts of which you've already seen being mentioned in previous talks, so I'm hoping I don't have to motivate the idea too much. So for reference, this is also, I'm going to talk about how I got here with the intent of communicating that I've been learning stuff that went into my dissertation research ever since I was in the undergrad program. So I grew up in India. I spent 5 years learning statistics in first in Xavier's College and then ISI. Mostly to do with probability theory. There was some linear algebra, there was some basic statistics and computing. For my graduate degree at ISI, I learned about Bregman divergences. And it turns out that Bregman divergences are very closely related to information theoretic notions, which is stuff that I've worked on for my PhD thesis. In addition to information theory, I've worked on causal discovery problems and a little bit of non-parametric statistics as well. So in this talk, what we'll do is we'll go over some association studies in statistics, and then we'll talk about association from an information theoretic perspective, where the key measure of association is called mutual information. And then we'll talk a little about challenges in estimation and inference, and key areas where my research has been helpful in that area. And then finally, an area of application, which happens to be reconstruction of gene networks. So typically, we've seen already that measuring dependence in data is a key issue in statistics. Some typical measures of correlation, which you've already probably seen, include Pearson's correlation coefficient, Spearman's rho, and Kendall's tau. And these are stuff that you've probably seen and also maybe worked on in your undergrad intro to statistics classes. These are popular measures, but they're also not great measures. There are some pathological examples where these data points, where these measures fail. Right, so there are good data sets where you can capture association using Pearson's R, Spearman's rho, or Kendall's tau, and there are pathological examples where these measures fail. So the ones on top you'll see are very typical examples of scatter plots that you'll see in your Intro to Statistics data textbooks, but there are also data sets where scatter plots might resemble the patterns you see on the bottom row. And these are typically bad data where Spearman's rho, Kendall's tau, and Pearson's R fail to capture association. As you'll know, the first three measures, they either focus on moments of random variables or ranks of random variables. Whereas the measure that I'm going to talk about, mutual information, deals with the densities of these underlying random variables. Using information theory, mutual information is able to capture or detect association in all of these cases. So that's a step ahead from the measures of association that we've already seen. But before we move ahead, I want to talk a little about mutual information and information theory. I think that information theory is this really nice mix of probability theory, statistics, and engineering. Information theory also has a really nice connection to Michigan. It was formalized by Claudie Shannon, whose photo you see here. You'll see some very popular names: George Boole of Boolean algebra fame, Alan Turing of I keep forgetting the name of the movie, but from cryptology and number theory. And the measures that are very popular from information theory that you'll see from any basic textbook would involve entropy and mutual information. So Claudie Shannon happens to be a Michigan alum. He went to undergrad here, which probably makes it more obvious why he named mutual information as MI. He proposed this when he was in MIT, I think, but I guess he wanted to pay homage to his hometown. He was born maybe an hour from here, I think. Mutual information is a really powerful measure of association. It's not only able to detect correlation, it's also able to detect independence. If you've taken an intro to a stats course, you'll know that correlation implies association, but association doesn't always imply correlation. And mutual information is able to detect the harder case of association and not just correlation. This is the only mathematical formula I have on my slides. I have no more. Mutual information between two random variables with a joint density on the numerator and marginal densities on the denominator. That's how you define mutual information for those two random variables. You can use this quantity to test if two random variables are independent. And then if you do confirm that they are dependent, you can also sort of estimate a strength of association between those two. There are some challenges in estimating mutual information. The existing methods are kernel density estimation or k-nearest neighbor-based estimation. Again, if you've had exposure to statistics classes before, you'll know that these are typical non-parametric estimation approaches. And you'll also know that typically these non-parametric estimation approaches have some sort of tuning parameter in the estimation process. And what happens there is that you have to choose a tuning parameter before you start your estimation process. And therefore, the answer you get is going to be biased by your tuning parameter. There's a lot of work that addresses this issue in terms of grid searching over a specific range of tuning parameter values, but eventually, regardless of what measure you take, the output you get is indeed going to be dependent on a tuning parameter. So one of the major areas that I wanted to work on was, are there tuning parameter free areas by which we can estimate mutual information? So that has to be one of the requirements that I had. The other two were, is it able to indeed estimate mutual information better in terms of having better accuracy, reduced variability than these existing methods? And third, can I provide some sort of theoretical guarantees in terms of how badly this estimator will do in a specific setup? Can I talk about quantifying how well it's able to test independence between two random variables in different mathematical settings? And it turns out that the product that came out of my first thesis chapter is indeed able to do all of these. What's even better is that it's able to do this with a massive reduction in terms of computation time. These are all great things from a research perspective, because then you get to deal with larger and larger data sets. You get to deal with more diverse areas in terms of being able to derive meaning or information from data where there was none before. With that, I'll move to an application area, which happens to be construction of gene regulatory networks. This data and motivating example have been taken from a 2012 data paper, although there are more recent examples of this. It's just that this paper seems to have really cool images, which is why I've chosen this one. As is always the case with any biostats research problem, there's going to be two areas that you have to focus on. One, the scientific problem, and two, the statistical opportunities and challenges. In what is a very reductive attempt to explain gene regulatory networks, so these are intricate systems of genes and proteins and other molecules, and these systems indirectly or directly determine to what extent genes are expressed in an individual, in the sense that whether these genes are turned on and off. And what that implies from a biological perspective is your gene expression is also going to influence how certain phenotypes or certain physically observed traits are being expressed as part of your biological data. So in terms of looking at the image, you'll see that the gene regulatory network is sort of like the network you see on the left of this image. What they do is they give rise to gene expression data. Now, we don't really know what the network on the left looks like. What that means is we don't observe the gene regulatory network. What we do observe are the products of interactions of these networks, which yield observable gene expression data, which happen to be the matrices you see on the right of this plot. So what we really have to do is reverse engineer or create reverse pictures moving from the gene expression data matrices you see on the right and move to gene regulatory networks on the left. And we're going to do this using mutual information. So what we do is, as a proof of, this is entirely a proof of concept. So we know this 10 gene network from the DREAM3 dataset. So the image you see on the left, or the network you see on the left, is the true network which gives rise to gene expression data, which are the matrices you see on the right. So what our aim is going to be, again, is going to take these matrices that we see from the right-hand side of this plot and try to reverse engineer our way back to this underlying true gene regulatory network. What we do is we're going to use mutual information to identify regulatory relations in this network. So what you see right now is sort of like an intermediate reverse engineered network. And you'll see that there are some lines which are not solid, which are dotted, and also I've shaded them in red. So these are the wrong guesses that the first round of your algorithm is going to yield. What that means is that your algorithm has probably not stabilized, or it's not reached a solution where it's completely in a stable state. So what that means is you have to keep running your algorithm until there's a certain level of stability or convergence. And that's exactly what we do. We try to fine-tune the output of our algorithm, and we get to a fine-tuned reverse engineer network. If you want to compare the plot you see on the extreme left with the plot you see on the top right, you'll see that there's just one guess that our approach gets wrong. And that's the node, or that's the line connecting gene 9 with gene 2. Everything else was properly captured in this approach, which is a reasonably good proof of concept of this idea. Why this matters is because our method, which is using mutual information, is highly sensitive to nonlinear associations, which is something you'd expect in these gene networks. Also, because we're computing mutual information of genes on a pairwise level, this approach is massively parallelizable. What that means is you don't really have to run a pairwise comparison of two genes one after the other. You can speed them up if you have a cluster. Michigan has a great cluster, by the way. So in summary, what we've done is we've spoken about association studies and statistics, and we've spoken about how there's a need to find measures which are able to detect not just correlation, but association. Then we've spoken about association from an information theoretic perspective. And we see that mutual information happens to be one such candidate measure. We note that it needs a good estimation strategy, which is something that we deliver on through this FastMI-based measure of mutual information. And finally, we see a proof of concept where FastMI was used to infer gene networks. With that, these are the references that I've mostly relied on while working on these slides. If you want to talk more about my research, please feel free to scan that QR code. I also have my website on. And with that, I'll be happy to take more questions. Thank you so much.
As usual, I have a question. Sorry. So you say that the FastMI is like O log n or something instead of ON because it's on the fast Fourier transform?
I don't quantify, so I haven't computed time complexity in the form that you're asking for. What we do is we provide empirical evidence in terms of comparing how our data set does, or how our estimation method does in comparison to existing methods. And there's a huge time.
Because you are using the fast Fourier transform, right?
I am using the fast Fourier transform, yes.
Oh, thank you. I'll take a look at that.
Cool, sure thing.
How did you decide to do like gene expression research? Like what drew you to it? Because like biostatistics obviously can apply to so many things. So what drew you to it, I guess?
Thanks. So first off, full disclosure, I didn't start working on gene expression data to begin with. I was more interested in the statistical problem in terms of being able to quantify or estimate mutual information well, because that's a problem that I sort of knew about as I was coming into the program, and that's the focus that I had. But while I was working on this, it's been great to know that there have been approaches where they're using mutual information to infer gene networks, for example. I couldn't add more in the interest of time, but there are tons of other examples where mutual information is used even today. So that's how I sort of reached towards areas of interest which sort of go in line with the biostatistics department program. Although if I were, let's say, in a stats program, I could talk about more of financial data stuff. And I think these methods would be just as applicable there.
I want to thank Jean and Soumik for two really nice talks that, besides showing you some of the kinds of things that people in biostatistics do, I think illustrate nicely several important points. One is just this notion of taking advantage of mathematics and statistics and computation to address really important problems in biomedicine, to take advantage of big data sets, to do that and be able to do that, to be able to handle the computation, to be able to take full advantage of the information that's available to us. And then one of my favorites, actually, that I have used throughout my career is stealing ideas from other fields and taking advantage of things that have been developed that you can then, by knowing about them, apply in a new and different sort of way. And that is an incredibly useful thing to be able to do.
All right, we're going to get started again. Good morning. Thank you all so much for coming. We're so grateful that you made it out here on this rainy day. My name is Kelley Kidwell. I am a professor in the department. I've been here 11 years, and I am the PhD admissions chair. I had been the full admissions chair the last few years and on the admissions committee when Mike was chair for many years. And so I'm going to talk today about the admissions experience. Feel free to ask questions. So you already know what biostatistics is, I think, from the intro that Mike gave and then from our faculty and student presentations. But I just really want you to know that we're among the the best in the world. Our graduates obtain really well-paying, interesting jobs. One thing that's really great about here is that there is just this update to the tuition and stipend. So you definitely want to think about where you do apply and how much that money goes, how far that money goes. And here it's quite a good deal now. So definitely think about all of our PhD students are fully funded. A few of our master's students are supported either fully and/or we have many tuition scholarships. So this is just a really phenomenal degree, and our degree combines theory, methods, computing, and application. And I'm going to talk about the different degrees that we have and the differences between them. So we have many accolades, and we're the only biostatistics department who ranks less than 5 in all of these categories. So this is really just a phenomenal department. It's a large department, but I think that's really one of its strengths. Any kind of area you want to go into, we have a faculty member here who is an expert and just usually world-renowned in that area. So you don't have to know what you want to study. You can come here and find out, and someone here will do that. We have 259 residential students, so our program is residential. Classes are in person. That's split 157 master's students to 102 PhD students. We have 44 faculty and over 2,000 alumni. So not only do you have a great cohort of students when you're here, but then once you leave, there's a massive cohort of students who have gone through our programs and are excellent connections to the career world and can really help you there as well. We also have phenomenal research funds. Our faculty are highly devoted to research, and that usually supports our students, especially in the PhD program, and provides really interesting work and dissertation projects. We're part of the Greater School of Public Health, which has several departments for which you can take courses across any of these departments and also across all of University of Michigan once enrolled. So if you want to take a statistics course or a bioinformatics course, those are all options as well. So in terms of the application, we have a holistic admissions process. So we're looking at your math background. You know, we have these prereqs of multivariable calculus, linear algebra, and an intro stats class. But we're also looking at, so we're really focusing more on that math specific GPA, but looking generally at your transcript. But we're not just looking at GPA, it's more but than that. We're looking at your letters of recommendation and your statement of purpose and personal statement. So we love to see intellectual curiosity. We love to see interest in public health and biostatistics. We love to see something about our program, especially for why you're applying to us. And then if you have other mathematics courses beyond our prereqs, that's great, but they're not required. Also notice that there's no biology requirement. There's no specific computing requirement. All of that we anticipate that you will learn once you get here and enroll in our program. So I have a few tips for you all, one of which I just stated. So when you're applying, we recommend that in your either statement of purpose and or personal statement, you make it clear to us on why you're applying for a biostatistics degree, whether a master's or a PhD. So why biostat? Why not statistics, or why is that not bioinformatics? What is it about biostat that really interests you? And then why are you interested in University of Michigan? And I suggest for, you know, you're likely going to apply to many programs, you have some sort of at least one paragraph that you tailor to each individual program that you're applying to that explains why that program. Is it certain faculty members? Is there a certain event that you went to? Is there a certain student that you talked to, or particular opportunities that you know about that program? Describe your background, of course, your current and future goals. You can specify faculty. It doesn't lock you in if you specify faculty or research interests. So don't worry if you write something in, we're not going to hold you to it if it changes down the line. You can just let us know about your interests now. Write clearly and well. So we recommend that you get others to look over your application to give you feedback. Now with AI, I imagine that AI can help you with that as well. I just recommend that if you do use some AI software, that, you know, it's really still about you, not about some generic interest in biostatistics. So it's more personalized, and you would have written it had you been able to have that literary expertise. In terms of your letters of recommendation writers, please choose letters who know you well, letter writers who know you well. So not just that you were in their class and you got an A or A plus, but there's more than that. You went to their office hours or you did a project with them, or they can really comment on your potential and your curiosity or your work ethic. That's really what we want to see. We also really want these letter writers to be relevant to the program. So, math, statistics, computing, research experiences. It's great if you had some sort of volunteer opportunity. That's wonderful. We want you to live your life in that holistic way, but it's not really pertinent to us. So we don't want that letter writer, but we hope that you do have those experiences. If you have any blemishes on your transcript, such as like maybe the first year was really tough, or there was some transition that was rough, or some course was really difficult for some reason, it's better to point that out in your application, either when you're filling in information about your courses, there's a little line where you can write some things, or in your personal statement or statement of purpose. Just let us know what happened and that it maybe wasn't representative. You don't have to go into too many details, whatever you're comfortable with. But it's better to address it and to not just sweep it under the rug. We will notice. And so what are the important parts of the application? I can't really point them out. It's all important. This is a holistic process. We're looking for the potential in you. And so we're looking at each individual application and really thinking about how much you're interested in biostat, how well you'd fit in here. So everything is important. Do your best on the application. So a little bit about our admissions in terms of numbers. This is from last year's admissions process for those who entered in 2023. So we had 945 total applicants. This is quite a popular place to apply to. There's a little bit of redundancy in some people being applying to multiple degrees, but you can see that it's about 2/3, 1/3 split between our master's programs and our PhD. It says MPH, we no longer have that program, so you can consider that between our two master's programs. So the applications were quite high, over 900, and we ultimately matriculated. So this is not the number of acceptances, but the number of students who came in the fall. 74 master's students, of which 2 were fully funded and 6 had partial funding, and 30 fully funded PhD students, of whom 18 came from our master's program. So they were second-year master's students in biostat and applied to our PhD program. Just a look at what individuals have previously studied. So when I walked around in the morning, I heard many different backgrounds, which was quite interesting. I actually didn't hear a math or stat background, although I didn't talk to everybody. So a large portion of our students come from a math, stat, or biostat background. However, we have a lot that come from a biochem background, some engineering, some computer science, business or economics, and then some other individuals with different backgrounds as well. So we're while we would like to see those prereqs, and those are most common in sort of a math stat background, we really appreciate the diversity of backgrounds of our students. We believe that the heterogeneity of the student body really adds to the, really great thinking of pushing the science forward. So we really like to see these different backgrounds. Our PhD students are a little bit more focused. Many of them are coming from our own masters. So you can see that primarily they're coming from a math stat background, although we have some from a specific data science background. And we also this year had someone from a finance background. We talked a little bit about our wide range of research areas, but I just want to let you know that, again, one of the advantages of this department is the wide breadth of opportunities that you have here, that we not only have individuals who are experts in genetics and genomics, but we also have people who are experts in computationally intensive big data analyses, in imaging and survival and survey methods, clinical trials, Bayesian methods, infectious diseases, many, many more areas. So whatever you're interested in, someone here is an expert in it. Okay, so on to the degree information. We have 3 main degrees. This would be our second year of our master's in health data science degree. Our master's in biostatistics has been around for quite a while, as well as our PhD in biostat. And I'll try to make it clear what the differences are between these degrees. So our master's programs, both of them, are 48 credit hours over 4 semesters, right? This is like a 2-year program. You generally do not take courses in the summer. So it's over the fall and winter semester in 2 years. about at least 36 of those credits have to come from our biostatistics courses. Many of those are core classes plus some biostat electives. We do require 3 hours of an epidemiology course and a one public health module or public health science course. And then of course you can take other courses, other electives across any number of areas. So you can take them more in biostat and statistics and computer science and epidemiology, et cetera. We're really focusing in our master's program to make you a wonderful team member. So we're giving you all that foundational statistics basis and really teaching you how to be an effective collaborator, communicator, and data scientist. In our Masters of Biostatistics, this coursework has as core courses our probability theory and statistical inference series, a statistical methods or applied regression series. And then instead of having a test at the end of your master's or a thesis, we have this kind of more capstone course, which is called Biostat 699. That would be taken at the end of your second year. And it's the analysis of biostatistical investigations. You get data, you get projects throughout the semester for which you analyze the data and present on your findings. So it's a really great way to bring everything all together that you've learned and to apply it and to get ready for what you'll be doing in the workplace. And then of course we have a number of electives, both in biostatistics and outside of biostat. You can go onto our website and look at the coursework and the courses offered. Our health data science master's has several of the same core courses. However, they have some different core courses as well. And the master's in health data science is going to still give you that great statistical foundation, but it's going to focus more on computing and big data. So there's a little bit more of a focus on programming and computing, although of course you can take any of the classes in any of the degrees. Okay, so the PhD program individuals can take these courses, the master's in biostatistics individuals can take these courses. So it has some new courses in data management, computing, model analysis, and interpretation. And this degree is a bit more targeted to students who are interested in a professional degree, so who would subsequently go into the workforce. Whereas our biostatistics master's is also a great professional degree, but will have some prereqs that are setting you up for also applying to a biostat PhD if you're interested. So some of the core coursework is the same, like our probability theory core and several of our regression or applied core courses, so 601, 602, 650, 651. But then there are several new courses for which are core in the health data science coursework. And those again are going to be focused on computing, algorithms, computing with big data, introduction to health science, machine learning. And instead of taking 699, that capstone course that the biostat MS students will take, the health data science master students will take what's called 629, which will be a similar capstone course, but it's case studies in health big data. Our PhD program, so the master's takes 2 years, right? And our PhD program is typically 3 to 4 years post-master's. So if you're applying directly from undergrad, this would likely take you 5 to 6 years. And if you're applying directly, if you're admitted directly from undergrad, you will essentially take our master's coursework for your first 2 years. And then you would move on into the more advanced PhD coursework. So you take the master's coursework and then about another year of courses, which includes advanced calculus or real analysis, a statistical inference series and stochastic processes, and then a number of additional higher level biostatistics electives. We do have a qualifying exam for our PhD degree. So after if you come in without a master's, after the 2 years of your master's coursework, you take the qualifying. If you come in with a master's, you might take this qualifying exam after 1 year of coursework, depending upon your what you've taken in the past. And this is one qualifying exam, which is testing your knowledge of those core courses from the master's programs, the probability theory and the applied regression classes. In our PhD program, you will be working on a dissertation. Often in our department, we focus on 3-chapter dissertations, which are usually related, and you work with a faculty member and or a few faculty members as chair or co-chair, and then a larger dissertation committee on that dissertation. We're really focusing in the PhD program to give you the skills to be a team leader. So the master's is to be a team member, and then you can become a team leader when you're out on the job and learning those skills on the job. But here in the program, we're going to focus not only on the team member, but also the team leader skills. A little bit about the funding sources. So again, all of our PhD students are fully funded. So that means that there's stipend, there's tuition paid for and a stipend. And in exchange, you either have a graduate student research assistantship, a graduate student instructor position, or you might be a fellow if you're a US citizen. So 94 of our funded students were research assistants, and they work kind of more like an apprentice with a faculty member on either applied and or theoretical methodology work. 22 of our students were graduate student instructors, and so they're teaching assistants for our biostat faculty in either biostatistics courses that are taught to other members of the School of Public Health and or to our biostat students. We have 2 training grants, which I'll talk a little bit more about in the slides that follow, a genome science training program and a cancer biostatistics training program. And then we have a few other fellowships that are offered from our graduate school, like the Rackham Merit Fellowship. And fellows are supported to take courses and to do research. And then we have a number of tuition awards. 12 of those were awarded last year, which are somewhere between 1/4 to 50%. This year, we might be offering full tuition to some students. And those are particularly for our master's students, since our PhD students come with that full funding. So a little bit more about the genome science training program, for which if you're interested, you should talk to Mike at lunchtime. But this is a world leading program in its 28th year. It's quite amazing. It's an NIH funded training program that's an interface between biostatistics, computation, and genetics. And this supports 13 pre-doctoral trainees. There are 49 faculty across 9 departments, 11 of whom are in biostatistics. So this is, really an interdisciplinary training program, which makes it quite strong. If you're interested in this program, you should include it in your statement of purpose that you're interested in this program, and you might be contacted to apply to provide another essay of your interest. The ideal background for individuals here is to not only have the math and computing knowledge, but also some more biology and genetics knowledge as well. So sometimes students start early on this program. There's an additional seminar series and journal club, orientation, annual retreat. They have funding to support meetings and extensive mentoring and tutoring. So it's just a phenomenal group to become a part of immediately upon coming to campus. Along with the genome science training program, we also have the cancer biostatistics training program. This is a little bit smaller. This supports 4 PhD or PhD bound students funded by the NIH. And this is really for exposure to cancer research for biostatistics students. So there's the biostat coursework plus some expanded cancer specific research. And then there's a journal club and funding to attend meetings that comes along with this as well. There are very strong links between some of our faculty members, those that lead this, like Vera Belladante Thupani and Jeremy Taylor and Phil Boonstra. Very strong relationship between those and others, and the Comprehensive Cancer Center. So you get really great relationship with individuals working on cancer research. The best background for this is a math or stat major and interest in science, although that's not to say that if you're not a math or stat major, they're not interested. Please, again, if you're interested in this area, put it in your statement of purpose so we can reach out to you in the application process. I'm going to let the students talk a lot more about StatCom, but I just want to let you know that this is an awesome resource that we have, student-led group on campus. That's a community outreach program that the biostatistics students actually lead. And so I believe the student panel will talk a lot more about this and tell you, but this is something that's somewhat unique to Michigan, although there's now been different programs that have popped up across the university or across different biostat departments across the nation, partly based on the group here giving support and guidance for those. So it's a really cool opportunity to do some research and to get experience and to give back to the community. So with that, I want to leave you with the timeline. So applications are due December 1st. If you are a senior and you have not yet asked individuals to write your letters of recommendation, that is your homework for today. Go back and ask those individuals, let them know that you're really interested in applying and that they should get their letters started. We will start to make decisions as the applications are completed. So you will definitely hear by mid-February at the latest, probably maybe late February at the very latest, but we generally have, if you haven't heard from us before that, we have a big funding meeting in February. So we'll let you know definitely by then. The decision deadline for you to tell us that you're coming here is April 15th. And then we send a form to you all asking about your interest in funding. Would you like to be a TA, an RA? Who do you want to work with? What courses interest you? And then we do this matching over the summer of what your interests are and what faculty have opportunities, and try to give you your, one of your top options, and also our faculty their top options. We'll have the new student orientation in late August. And of course, you'll start with the start of the new school year, which the last time I looked wasn't on the registrar's page yet, but it's sometime around like August 20 something. So I'm sure that will be the same. All right. If you would like more information, please see our website. We have an awesome frequently asked questions page, a fact page that we spend a lot of time on for admissions process. You're also welcome to email any one of us on this, Nicole, Fatma, who is outside, or myself. And I will also open up to questions now. So any questions? Yes. And remember, yes, turn on your mic, please.
I guess this is kind of specific to me, but like I'm already working as an RA. Like, does it have to be specifically biostatistics related if you want to work as a GSRA?
Oh, okay. The question is, right, does it have to be biostatistics related? So usually the research assistantships are like assigned through our department. So it's possible that if who you're working for would like to continue funding them, that we could ask them and put that kind of information into our funding information and help use that as a match. But primarily that's a full departmental process, so that it can't come from like you initiating that. Nicole, do you want to add?
I just want to add that there was actually one student who was working with somebody and we were able to take that information in and input it into the funding matching scheme. So if you have a connection with somebody, just be sure to list that in your application and maybe follow up with me. And then if you're admitted, then we can try to work something out with the person who's funding you.
Did you want to follow up, or is that? Okay. Yeah, just let us know and we can follow up with it. Any other questions? Yes.
Hello.
Oh, just 1 second. Let me get one in the room and then I'll go to online.
On average, how much is it going to cost the entirety for like a student?
Like if you get a master's, how much does the program cost? Nicole, do you know those numbers? It depends if you're in state or out of state. Are those numbers on the Rackham website? So no.
I would look at the registrar's office and look up the public health program and Rackham students. It doesn't really matter. All students are charged the same tuition at public health, but it does matter if you're in state or out of state. And then the amount changes. And so I don't quote it anymore. I used to, but I can't.
So yes, I know how much it says it's going to cost, but you said like student matching and things. What's the average?
So if you are fully funded, then you don't pay anything besides like your living expenses, right? But hopefully our stipend is covering that. If you get tuition support, it could be anywhere from 25 to 100% tuition support. And otherwise, if you don't get any funding, then it would be you paying for the degree. However, you can also talk to Rackham about financial aid.
Yeah, I noticed you didn't mention the accelerated master program. For that program, is the timeline going to be the same? That's my first question. I have another one if you don't mind.
Yes.
Okay, so my other question is like, what percentage of students are doing like direct PhD in the PhD of biostatistics?
Okay, great questions. Let me talk about the first. That was an oversight. We should have talked about the accelerated master's program. That is something that is new for this year for University of Michigan students, that we have something called the accelerated master's degree program, for which as you're a junior, you apply, and actually the application is not due at December 1. It is due later, I think up until March possibly, but you should look online. We have a section, or we should, if we do not, we'll make sure that's there this week. So you apply in your junior year to be a part of this program, for which you would start to take our courses your senior year and then finish out degree the year after. But there is a extended period of time for which you can apply to that. And then you have to apply again officially to Rackham in your senior year for our master's. The second one was the second question about direct PhD. So direct from undergrad to PhD. Nicole, do you know the percentage? Yes. You can send me an email and I am happy to follow up with that number. I know that we have it somewhere. I just don't have it off the top of my head. What I can tell you is that we had 18 internal individuals who definitely had their masters. So, and we had 30 total. So it's definitely less than 12. I think it's somewhere around 5 to 9 of the external individuals just had their BS or BA, and a few other had a master's. Great question. So no, you do not need an advisor before your PhD program. And as I said, you can put faculty member names in your application and we won't hold you to it. Again, we'll figure all this out through the matching process, at least who's funding you in the PhD program. But that doesn't mean that that person has to be your dissertation advisor. So you may be funded initially by someone, and then in your time through the program, as you're going through courses, you realize your specific interest and you can reach out to that faculty member, and that can be a dissertation advisor. So we don't accept you as like an apprenticeship model. That's a departmental process and decision that you're accepted to the department. And then once here, we think you will be able to find that individual, and you have flexibility in that, because who funds you doesn't necessarily need to advise your dissertation.
Sorry, it's kind of like a follow-up of the previous question, but would you say it's like much more difficult for an undergrad to do a direct PhD rather than getting their master's and do PhD?
We are very interested in all applicants, whether they have a master's or undergrad degree. If you have those prerequisites, we definitely consider you for the PhD. I think sometimes it might be somewhat harder to see your research potential as an undergrad. However, it's not impossible. And so we have definitely accepted individuals from undergrad. And we think that if, you're truly interested in the PhD and you're going to work hard through the program, you can definitely do that very well. And you don't need to go through the master's to the PhD. It's your sort of your choice. I guess the other thing I should tell you is that if you apply to the PhD, so if you're interested at all in a PhD from undergrad, you should apply to the PhD. And then you will be considered for the PhD. And if we decide not to admit you to the PhD, you'll also be considered for the master's program. And there's a checkbox on the application where you say, please consider me for the MS in Biostat, or please consider me for the MS in Health Data Science. And so we will automatically include you in our master's decisions as well.
An example, I am from I have done my master's in biochemistry and I wanted to pursue a master's in biostatistics. So in that case, when I'm up, the prerequisite for the courses, I have to complete 3 courses of math and 1 introductory courses of statistics, which I am doing. But my question is, like, if I am if I registered and if I'm not completing before December 1, so am I eligible to apply for the program?
Oh, yes. You do not have to have fully completed the prerequisites when you're applying. You should let us know that you're taking those. And then we sometimes offer what's called a conditional admittance, where we will say, you will be admitted as long as you get like a B+ or above in linear algebra or whatever, if that's the last thing to take. So then we'd ask you to send us your grade.
if it is from a professor or somebody from where I was doing the math course, or if it is related to math or statistics course, maybe there are three letters of recommendation is required. Maybe I can have one or two from like mathematics and statistics field, but because I have done biochemistry and I have research experience, can I put in recommendation letter from that field as well, or that will impact my application?
Yeah, just as you said, you have 3 letters of recommendation. So, all 3 do not have to be math and stat. It definitely makes sense if that's your background to have that, but it's great that if you have research experience to have, 2 maybe more math or stat and 1 from that research opportunity.
Oh, thank you.
Yeah, thank you. And if you still have questions, please feel free to put them in the chat or to email any of us on screen. Unfortunately, I can't stay for lunch because I'm going to coach my daughter's basketball first game, 7-year-old. So wish me luck. But I'd be happy to follow up in email. Thank you so much.
Hi, everyone. My name is Mike Sweeney. I am a 3rd-year PhD student in the department. I am a current trainee of the Genome Science Training Program that was mentioned before. I was directly admitted to the PhD for my bachelor's. I'm also a first-generation college student, and I work in statistical genetics with Dr. Hyunmin Kang in the department. I got my BS in computer science and applied math from Stony Brook University in New York in 2021.
Hi, everyone. I'm Hannah. I use she, her pronouns. I am from North Idaho, and I went to a small liberal arts college in the Seattle area for undergrad. I am a 3rd-year student in the department. I got my master's here first, and now I'm starting in the first year of the PhD. And I am interested in clinical trials, and we'll be working with Kelley Kidwell starting next semester.
Hi, my name is Suyeon. I'm from Korea. I had my bachelor's in statistics at the University of North Carolina, and I'm a first-year master student in biostatistics here. So let me know if you have any questions.
Hi again. I'm not going to waste your time and introduce myself again. I work on causal inference problems and causal discovery problems. That's the only thing I didn't mention on my slides. Please let us know if you have any questions. Yeah, we can talk to you about student life. We don't have to talk to you about research. You can ask us about the kind of food they serve, places we go to, whatever is good.
Hello. So my question is, I guess it's kind of simple, but I'm interested in the MS in biostatistics. And just like, what does a week of classes look like? Like, how much time are you actually in class versus studying? Yeah, if you are a TA or RA, what does that time commitment look like?
Well, so I will say the master's program, I guess, and the PhD by proxy, it is decently course heavy. You will be taking, I don't know, probably about 4 classes a semester. Do you think that's right? 4 classes a semester. There are 2 theory core classes and 3 applied classes that are in a sequence. So you'll take those in your first year and then your first semester of your second year. And I think on the first day of classes, I was told that per credit hour, you should be spending 3 times that number of hours on homework outside of class. I think that was a little bit of an exaggeration, but it is more intensive, I would say, for sure, than an average bachelor's experience, just for comparison, because the depth of the material is more. So you're still moving at a potentially a similar pace to what you did in bachelor's, but you're digging a lot deeper into a lot of the topics. And then for a research or like a GSI or a GSRA, those commitments, I think the on paper, it's 20 hours a week. It fluctuates a little bit depending on the given week or what's going on. Like, for example, if there's a conference paper deadline coming up, you'll be working more potentially than 20 hours a week on your research, but that's not, again, it fluctuates. So it averages out probably to that amount. And then with a GSI, I would say also it's about the same in-person time. It depends. You have to go to the classes. Sometimes you have to hold office hours, and then teach the labs usually. And then outside of that, doing grading and things like that. So I'd say it's a balance of all those things, but you still definitely have time, at least in my experience. I made time to do fun things. And I think that's really important in grad school to keep your sanity. So it is definitely balanceable, but give yourself grace in that first year for sure, because it is sometimes a learning curve.
I will also say, as a master's student, you're not really expected to make leaps and bounds in research. Your professor, like your advisor, will be understanding that you're taking classes, and since we are a coursework heavy department, they will be understanding that you have exams and homework. While there is a 20 hours a week requirement, as long as you're making progress, they'll be very happy with you, and they're pretty lenient about what they consider progress, considering you will be taking courses. Yes.
Connecting to that research that you just mentioned, how free would you say it's like hopping around to like explore different research opportunities? Would you say that the faculties are pretty welcoming in terms of like, can I try this out? And then they'll be like, yeah, sure. They don't really care.
So if you do have a funding or commitment from the department, where they talked about that matching form before, you will be those are at least a year-long commitment to that specific professor. They do match you in the attempt of building a long-term relationship with that professor. We do not have a rotation like other departments do, so you'll be working with the professor for at least a year, hopefully longer. But if you have a funding commitment, if you are a fully funded master's student or PG student, you do have funding. You can change at the end of the year. I changed myself after 2 years of working with someone. I moved to someone else. So you do have that funding commitment, but the intent is to build a long commitment between you and a advisor.
Also, to add to that, my first year here, I was working on some genetics problems. Just turns out it's not a good fit for me. So the department and I work towards finding a new advisor, starting a new research program with them. And I think, to answer your question, there were no bruised egos, so to speak. It was a really smooth transition from one research project to another, to the point that I don't feel like so this was after I'd already spent a year, right? So I don't feel like I really wasted a year to that point, even though I didn't work on those research projects as much. Because again, the expectation when you're an MS student or an early career researcher is that they're also going to teach you how to research. And there's really no textbook for that. And there's really no coursework for that. So they're going to be patient with you. I've been here for 4 years. I've seen on an average, let's say 50 students graduate each year, out of which, let's say 15 are GSRAs, right? These are really ballpark numbers. I'm sure it's more than that, but I've never heard anyone say that, hey man, the learning curve is really steep. It is steep because it's grad school, but I think it's reasonable in terms of the expectations your advisor has from you.
Yeah, and I'll echo too. I also change research. So I guess your sample size here is a little skewed potentially, but so in the master's program, I also worked on something different. I did mobile health research with Walter Dempsey. And we were doing hybrid trials, which was kind of a combination of mobile health and clinical trials. And I realized, you know what, I think I like these clinical trials a lot more than this mobile health for a variety of reasons. And he was like, you know what, that's great. You know this now. And then I was able to shift pretty easily to working with Kelley instead. So again, they're about trying to make this a good experience for you and making sure that for a dissertation, you have to love the topic you're doing, or at least not hate it, because you're going to be spending a lot of time with it. And I think all of the faculty here understand that and are trying to make it a good match for you as well.
In addition to what she said, I also think there are issues where a student's co-advised by faculty members. So if you think like you're working on an area which requires input from a specific faculty member for one project and another one for chapters 2 and 3, for example, there are students who have co-advising situations, and I don't think those theses come out worse than any others. So the department's really flexible. I can hold on to that, sure. More questions? Yeah, sure.
What programming language is most common in the master's? Is it R, C++, Python, and how much prior experience should you have within each of the softwares?
Can I start? So mostly it's R. I think about all of the courses that we have, barring one, the primary coding language is R. That being said, I don't think instructors put a great deal of emphasis on what coding language you're using, as long as you're meeting the deliverables. That being said, if it's a course about where you're learning how to draw plots in R, you'd have to use R. But apart from that, there's really no hard and fast restriction. But I feel like if you're not coming in from an engineering background, it's more likely than not that you'll have more experience with R. And I think that's sort of what's reflected in incoming graduate students' pools as well.
Yeah, so I also think that we use R the majority of the time and maybe Python with R. So, but you don't really need prior experience, I think. You have the basic courses that you can take that are like 607, the fundamental Python, R, and C++. So you really don't have to worry about that. Yeah.
Yeah, I would say background definitely helps, but it's not required, to echo that sentiment, because there's also Biostat 600, which is another useful class. It's like a way to catch up on all the, like, remind yourself of things. For example, if you haven't taken linear algebra in 3 years, remembering how that works is useful. So that course is useful because it covers, I think, linear algebra, R, calculus, and like basic probability, I think. And those are also good things to brush up on a little bit. And I think taking that course was useful because it prepared me, I think, a little extra for the classes that I was taking otherwise.
Regarding back to computing, Biostat 607 is a 3-credit course of R, Python, and C++. But the way that course is set up, it's split into thirds. So you can take one of those for like 1 credit. So you can take like all 3 for 3 credits. You can pick just the R1 for 1 credit. So that's a way to catch up on programming languages if you're not familiar with that. Yes.
What were the reasons why you chose this biostatistics program specifically? And then also, why did you choose biostatistics over, say, like statistics, which is basically the same field, but can be applied to more fields in general?
So I'll start. I have 2 main reasons. One of them addresses both of your questions. So I had undergraduate research experience in statistical genetics. So I knew I wanted to pursue that in my graduate degree. So obviously choosing biostats over something like stats or computer science was a natural decision for me to make. And then one of the reasons why I chose to come here is that this department has a really strong reputation for statistical genetics, as you've probably seen from the presentations earlier this morning, and a lot of people, that word comes up a lot. And I would say the second reason that I specifically chose Michigan Biostatistics is that when I would interview at different schools, I would say this department had a decided emphasis on compassion and also just like collaboration between everyone. I think grad school can be scary, and I think you want to have a supportive environment. And I think this department really delivers on that, and it makes a decided effort to deliver on that. And you have the wellness advocate, Shadima. And this department has a real commitment to making sure that this is a good experience for you, not only academically, but also personally.
Yeah, so in undergrad, I studied, I initially thought I wanted to be an actuary, and clearly I'm not there right now. So I did a couple internships and I thought the problems were really cool, but I was like, this is not very meaningful to me because I'm sitting at a desk, I'm not working with people, I'm not working for people. I need some more contact with people. And so I went to my advisors in undergrad at the time, and they said, oh, I think public health might be an interesting thing for you to check out. So I looked at statistics and epidemiology and biostatistics, and was kind of like figuring out what of those would be a good fit for me. And I really liked biostatistics because it's quantitatively equivalent to statistics, I would say. Like a lot of the classes here, for example, are cross-listed. So you can take courses in statistics and biostatistics for the same kinds of credit. And so I liked that it was still very quantitative, but biostatistics, I think, tends to attract people who care about people a little more than statistics. And I say this department reflects that really well. And then in terms of why Michigan Biostatistics specifically, like I mentioned, I went to a small liberal arts school for undergrad, and community was really important in that environment. And so when I was going to grad school, I was like, I definitely want to go to a place where I'm going to feel like I fit and people actually care about me as a human. And like Mike was saying, Michigan really delivers on that. when I applied, I mentioned Kelley in my application, and I didn't know this at the time, but she was the admissions student that year. And she called me and said, "Hey, we're considering you for the PhD, but we also have this funded master's. Is that something of interest to you?" And I was just like blown away at the fact that they noticed. Okay, because they have, a bunch of people on the admissions committee, and they took the time to say, okay, this person mentioned this person in their essay. This is going to be meaningful to this person. And all the conversations I had with students and faculty and staff were all really positive experiences. And so it really just fit all of those things. Plus, Michigan is obviously a really renowned research university. So it really does kind of balance those two things that I was looking for in a really positive way.
No. Sure. Okay. They took away all your answers. Is that it? I see. So I have a non-serious answer for you. So my father's got a PhD in statistics and my mother has a PhD in plant biology. So I had to make sure both people are happy, which is why I chose biostatistics. So that's a non-serious answer. But I think, again, I was tired of looking at integrals and differential equations all the time in my undergrad stats degree, also with my master's stats degree. And I felt like I wanted to learn more of coding. I wanted to learn more about, looking at data and being able to draw meaningful conclusions from them. I'm not saying that stats department don't do it. I feel like a biostats department's more oriented towards that goal. And as far as Michigan goes, I think, so here's what I think really worked out for me. I wasn't thinking about this when I was applying, is that it's a huge department. You have 42 faculty members here, right? So in a situation where, and typically when you're coming out from an undergrad program or even a grad program, you don't know the kind of problems you're going to work on. You'd be very lucky if the first project you thought about in your PhD program turned out to be the project you worked on, right? So in a situation where you start working on something and you don't like the problem, it could be because of it's not something you like working on, it's just too hard, anything, right? I think having a big department allows you to pivot to other research opportunities. And I think that's great from, making sure that your career is still on track in terms of meeting the right goals at the right time in terms of degree requirements. And also having a big department means if you're stuck on a problem, it's more likely that you'll have people to reach out to as compared to a small department. So yeah, those are the two reasons, I guess. Yeah, more. All right, sure, go ahead.
Sorry if this is like a more general question, but is there one course you wish you had taken, or like, is there like a skill you wish you have learned before you enter your program?
Sure, that's a great question. So I think this might be a person-specific answer. So I'll tell you what happened for me. 699 is sort of like the capstone course, which you have to take regardless of whether you're just mastering out or getting a PhD eventually. And it's sort of like a culmination of everything you learned during your first year or first 2 years in the program. You're given raw data sets to analyze, you're given some questions which might be of interest from a scientific or a medical perspective. And then you're kind of like given a lot of independence to play with the data and just look at cool methods which might be meaningful there. You also have to write a report which summarizes your methods work, your findings, and contextualizes them in a framework that's meaningful to clinicians and people who don't understand a lot of math. I think translating results from your R code to a meaningful paragraph is an incredibly hard jump, or at least it was the way for me because I had no training in scientific writing. So from the struggles that I had when I was taking coursework, I think that's the course, or at least some background in terms of scientific writing would have been really helpful. The other thing that I think was not like, because I come from a situation where we were taught a whole lot of theory and not a lot of application stuff, my coding skills weren't up to mark when I joined the program. I think you can take a lot of programs, a lot of courses, not just in our department, but also in the statistics department to level up your skills there. So I think you'll be just fine, even if you don't come in with those prerequisites. There's a lot of potential or a lot of opportunities to just learn as you go. It's going to be a more steep climb for sure, but I don't think it's going to be a really hard climb, All right.
So I I'm a first year student, so I don't know exactly what I have to suggest for you. But in my experience, I think having a stat background is pretty important. 601, 650 are, I think, one of the most hardest courses that you can take, and it's introductory, and yet it's quite difficult to deal with. So yeah, just be prepared to take those statistics courses. Yeah, that's all what I can say for now.
You'll be fine. You'll be fine. I guess one course I was glad that I took was real analysis. It's not required for the master's. It is for the PhD to have taken by the time you are a candidate, I believe. And so if you don't have that course taken already, you will have to take it here as a PhD student. And I do think having taken it in my undergrad helped me in the courses in the master's as well, because real analysis, at least the one I took, it was a lot of proofwriting and things that were, theoretical use of math in a more abstract way. And I think that kind of background helped me a lot in the theory courses here. I don't know if there's one I wish I would have taken. Off the top of my head, no, but I think real analysis was the one I was the happiest that I did take.
I probably have the exact opposite answer of Soumik. Just because, as he said, this is a very personal question. So I came here with a bachelor's in computer science, and I came here with like a first author publication. So like I was good on scientific writing and coding, but I hadn't touched multivariable calculus since my first year of undergrad. So I was like 3 or 4 years removed from that. And opened the 601 midterm and needed integration of my parts. And I was like, what the hell? And so I think in retrospect, I probably should have taken Biostat 600 with Hannah to refresh on that stuff, but I didn't. So I think that would probably be my word of advice. Or even if you don't take that course, if you just like really briefly review some stuff, like I think they send out an email of like stuff to review if you're not super comfortable with that. But I also wouldn't really worry about it too much. Yes. So all of our courses are in person. That's probably a different answer from everyone as well. So, the question was online versus in person, right? That probably depends on the professor. All the courses are in person. I think there's probably an expectation for you to show up in person. Would you agree with that?
If you do have issues in terms of joining in person, and if you're able to arrange something with the professor, all classrooms now have Zoom capabilities. So I don't see why having a Zoom option is going to be bad, but I don't know what the rules are right now, though. Oh, actually, yeah.
Yeah, I would say it's kind of complicated. So if there are
Yeah.
personal reasons that you need to, I think the faculty are pretty willing to work with students if there's a reason. The classes are sometimes recorded, and so you may be able to view the recordings later. Typically, it has to do with, some sort of sickness or health reason that you're not able to attend in person. But all of our classes are in person, so in-person attendance is expected unless there's some other reason. But we have very understanding faculty, and so they're always willing to work with students.
Yeah, so I'm taking the core courses. You can't really get the recordings for the core courses. You have to be in person. They changed it this year. I heard about that from the professor. So unless you have valid reasons why you can't attend in-person classes, you still have to be present, and they do take attendance for core classes. For other classes, I think they're pretty flexible. You can request for Zoom, or sometimes they make it hybrid for some cases, but usually like 90% of your classes will be in person.
Yeah, I think a lot of this actually is a result of the pandemic, because the first year that Mike and I were here, our core courses were online the first semester, and then transitioning back to being in person. They were really trying to make sure there were online opportunities for people because, you know, we didn't want people to show up on campus with COVID. So we had, you know, all the classrooms, like Soumik said, were fit with things like that would automatically record a lecture and it would go to Canvas. But then there were problems with people not going to class anymore because they didn't think it was useful. And then the professors got really salty. And so now they're not being recorded anymore. So I think, again, they will work with you if it's needed. But I think because of the pandemic, we had like, I don't know, 25% of our classrooms at capacity because people didn't think it was useful to show up in person anymore. And I do think it is going to be the most useful for you to go in person because it's just not the same otherwise. But I think, again, I don't think a lot of them are being recorded anymore. There are a handful that still are, but the expectation is that you are showing up in person. And a lot of them, again, because of faculty being upset about students not showing up, they do have attendance components now as well built in.
Warmth wise,
Yeah, if like how was it transitioning to Michigan weather? And like, are there snow days? Like, is that like from Florida?
I think we've had like one snow day in like the years I've been here. I'm from New York, so like I'm not that unfamiliar with snow. But I do live in an apartment complex, so I don't have to do any shoveling. And I do take the bus to school, so I don't have to drive in it. So I'm pretty happy with it. It is cold. My advice would be buy a winter coat, buy a hat, buy gloves, and I think you'll be just fine.
Yeah, and boots. Don't forget the boots. Yeah, I actually was underwhelmed by the snow here compared to where I lived. So I'm a little biased. But I will say it is cold and long. I think that's the biggest thing. The winters are long and it's gray in the winter. So that's something to mentally prepare for. But in terms of like bearability, it's totally fine. It's not that cold. It's not like you're going to have feet and feet of snow. It's not going to be like you can't go anywhere. You'll be totally fine. Like I still went on runs in the winter. But again, with some bias on where it came from. So yeah, I think with a coat and boots, you'll be totally fine.
Yeah, so for me, it's really cool. As you can see, nobody's wearing this kind of jacket. I'm wearing a puffer jacket. I spent last 10 years in a tropical area and I'm from UNC. So yeah, it's already cold for me and I'm not ready for that cold.
yeah, whatever they said. I was told that you need two things to survive in Michigan. I was told this by an Indian professor. One is a really good quote and two is a strong sense of denial. It's served me really well so far. So I don't see why it hasn't been a problem so far. So we should be good. Yeah. Yeah, question.
Yeah, is it possible that you guys could speak a little bit more to like the community aspect of this department? Like they mentioned some of the other, I guess, clubs or seminars that you could attend to. StatCom was something that they mentioned quite a bit during this presentation. What your cohort energy is like, the collaboration, whether you get to know them. Yeah, anything to that topic would be appreciated.
Yeah, I have a lot of things I can share about this. I'm over involved. I this year I'm a co-president of StatCom, like Kelley mentioned, the program, and Soumik is also part of StatCom on the leadership team. Though I guess you're sort of phasing out now, which is wild, graduating soon. But StatCom is a really cool program that she mentioned. It's statistics in the community. It's a way to use the things that you learn in the classroom in a very hands-on way. And you get questions from your community partner about these are the things we want to investigate, and then you go and do it. So again, it's kind of more of a free form way of practicing those skills rather than getting a data set and saying, oh, well, we're talking about logistic regression in class. I guess I'm going to fit that model. So it's a little bit more like real life, which is cool. And then also, you know, that outreach is a really neat thing. And it's cool to see the work that you're doing positively impact people. As you can tell, I like people and people are motivating to me. So that's something that I find really interesting. Also, I'm involved with the peer mentoring committee, which is a pretty cool program. So all first year students get matched with mentors that are upperclassmen, whether it's a second year master's student or a PhD student. And then that person is a really good point person when you're coming into the program, asking questions about coursework or about housing or whatever you need help with when you're coming and transitioning to moving here. And I do think the cohort being large is nice in terms of you have a better chance of finding your people, I would say. My cohort, I think we were also a little people starved because we were coming in right after COVID, but we all really hit it off right away. Like there was a group chat made before classes started and people were like, hey, I'm going to go get coffee at this place. I'm going to go check out this bar. Like whoever wants to come join, that's awesome. And so we really were interested about meeting each other. And I do think that is also a product of Michigan and biostats combined. I think people are generally excited about being around other people.
I will also mention that virtually all of the department committees have student representation on them. I am on the student recruitment committee. There's a brown bag committee. All those committees have students represented on them. Students can give their input and stuff like that. I'm also so is Hannah, an admission ambassador for the School of Public Health. There's DEI events and stuff that you can get involved with. There's a lot of ways to get involved that you can tailor to your particular interests.
Oh, wow.
Okay, so question online. All right, so I can give you figures from something I calculated earlier this year in March. So I don't have a family to support. I don't own a car. I mostly travel by bus or my bike if weather permits. And I split an apartment with 2 other people. So with that, my basic needs comes out to and I am a funded student. So with regards to my stipend, my basic needs as of March came out to about 30% of what my stipend was. I don't mean to say that I live a frugal life. What I mean to say is they pay us a decent amount where you can comfortably spend and also save a little. Of course, this is subject to my conditions in terms of not having, I stay off campus and it's like about 3 miles from the department. So I do have to take the bus. I can't really walk every day. So keeping those in mind, I think 30% is a decent percentage of your stipend to spend on basic needs. And I think comfortably all students save a little, unless you have like really expensive hobbies. So yeah, I think that's, and housing costs I think are typically between, if you are willing to split an apartment, off campus, I think they're between $600 to $1,000 per person, including utilities. If you're going to a single, Mike, do you have your own apartment? Yeah.
I also think that depends on where you're living, because that I'd say that range is more accurate. Like a North Campus across the river, I'd say that's pretty accurate. I live in West Ann Arbor, like I don't know how many of you are familiar with this. I live kind of near Blank Slate, the ice cream place. So I can walk to campus, 25 minutes walk. That's pretty easy. I can bike here in like 10 minutes. I can take the bus in like 15. And my rent is higher than that. And I'm splitting it with a person, but it's something that I was weighing and I wanted to be able to be more mobile.
Yes, as Soumik mentioned, I have a 1-bedroom apartment to myself. I live 2 miles south of the School of Public Health. By Briarwood Mall. My rent and utilities come out to $1,400 a month. But compared to New York, that's nothing.
And also if you live, you can also, some students do choose to live in Ypsilanti, which is, I don't know, 20-minute drive, 10-minute drive. It's not very far. So that's also an option.
So kind of in the theme of living and commuting, as much as I love Ann Arbor, I own a house in Southfield. So I would be looking at commuting. Is that something where you're like, horrible idea, don't do that? Or is it just, just like a job, just something you have to consider doing?
People do it. There are definitely people who do that. One of my friends actually just moved. She just got engaged. And so she's splitting, she is living halfway between where her fiance is working and halfway between there and here. And she just commutes. What she does is she commutes to a lot across the river. There's like a commuter lot. It's a cheaper parking pass that way, and then takes the bus into campus. So it's definitely doable. And I would recommend that format as opposed to buying a parking pass closer to campus, because parking is hard to find close to campus. And it's very expensive. So it's definitely doable. You'll just have to a lot more time to get here. Yeah, there's commuter lots. Yeah, park and ride, I think.
Yeah, it's like a park and ride, and the one by my apartment's free. There's no and you can take the bus to the Central Campus Transit Center for free with your University of Michigan ID card.
Do you want to, well, yeah, let's do one more question super quick, and then we can also answer more questions at lunch.
What is your favorite thing and least favorite thing about being a student in the biostatistics department?
Anyone ready for this? I need to think about it.
So I would say as a PhD student coming in, I was really interested in research. I do think this is a coursework heavy department. I don't think I had enough time in my first 2 years to really do substantial research that I wanted to do. That probably my least favorite thing. That probably doesn't apply so much if you're a master's student. But I would say my favorite thing is probably the community here. We have a lot of great scholars who go out of their way that are great academics, but also great people to talk to. We have faculty student lunches. Just this last Friday, I went to lunch with a group lunch with a professor. The faculty here do go out of their way to connect with students, and they talk about things other than academics. They talk about their lives, their children, stuff like that, their personal lives. And I think that's a really great humanizing way to, it's just a really nice community here. That's probably my favorite part.
Yeah, I think I would echo that the heavy coursework is probably my least favorite thing, because my first 2 years I was doing research, and I did end up getting a publication out of it, but like I did not put as much hours into that as I would have liked. And then my favorite thing, a non-serious answer, the department has a lot of free food. That is a big win as a grad student. But I will say also the PhD life is really flexible, which I really enjoy. So I'm able to really engage with research and do the things I'm here to do, but also can, take a long weekend and go somewhere sunny in the winter if I want to, for example, because research is something that you can do somewhat asynchronously as well if you need.
The favorite thing about this university is that I think it's a community, as what Mike mentioned. Yeah, so it's very diverse, and it's a very diverse environment as well. So even if you're an international student, you wouldn't feel left out, and the faculty is very welcoming as well. It's really easy to reach out to. So that's one thing that I really like about, and not really about this university. I just don't like the weather. So yeah.
This might not be a very general answer, but for me, I think there are some courses at a PhD level that are sort of meant to be advanced courses of the master's level courses. In my experience, I've seen that the jump between these is not an easy one, especially with the 600 level courses they were mentioning. 601 and 602 happen to be master's level inference classes, and 801 and 802 happen to be PhD level inference classes. I think that the jump between these is not an easy one. And over the years, I think it hasn't improved. It hasn't worsened for sure. But I think what I'm trying to say is that's probably the biggest thing that I feel would be easy to change, maybe, but it's still not there yet. Oh, I'm gonna lie about, I don't think you'll believe me when I say this, but there's so many things. I think the biggest one is probably that if you are, I know that this is a hard thing to say, but very early on in the program, I was told to ask whenever I needed help. And I think that's really good for advice on the face of it. More often than not, I come from a culture where it's more normalized to sort of work through it and sort of struggle a little before you go and ask for help. I feel like that's really time that you're wasting and taking away from your studies, from your friends, your society, your social commitments, and so on. So, and the department's great at providing support, but the caveat is you have to ask for it. Yeah. Oh, perfect.
All right. Oh, am I an adult? So thank you so much. And I'm going to stop the recording. And thank you to our online viewers. And that's the first time I've ever said that.